Okay, so what is the picture you are seeing here? Huh? Whole genome. Okay, so these are chromosomes, these are genome, and we are going to talk on genomics today. How many of you actually are working on genomics subject? Actual hands-on working on your projects? How many of you? So I think many of you will be working on genomics because in biological field in Bangladesh specifically and in other countries also, we need to work on this. Otherwise, we cannot explore many things hidden in the DNA. So genomics is not a single subject. You all know that it comes with it has a very close relation with genotyping also. So genotyping, microarray sequencing, these all comprises the genomics. And today we will be talking about some aspects of sequencing and the mainstream genomics, not the downstream one, the thing that generates the data. So thank you, CompBio Bangladesh, for inviting me to be a part of this project and delivering this lecture today. My name is Sabrina Elias, and I will be first talking about the genome sequence techniques. Right. If we do not know about the genome sequence techniques, I mean, how we actually do the sequencing, how we generate the data, then we will not be able to analyze the data, right? So when we know the data structure, then it's easier for us to analyze it. And the main charm of the genomics is it can capture the variation. What are you seeing in the slide? So it's a variation, right? We all are human. We all are 99.9% similar, but there is 0.1% at least difference. And genomics nowadays can capture those okay, sorry. So natural variation, I mean, in the section 3, you are seeing a genome-wide associational studies. Many of you are familiar with this, and many of you are actually doing this. But earlier days, we used to do the genome sequencing assembly, RNA-seq, but nowadays, the comparative genomics, which is the genome-wide association study or the linkage study, these are being more popular and these are being more functional. So, shall we revisit the central dogma? We may not need, but this is a favorite slide of mine because it shows that how we actually get the data. So it's from DNA to RNA to protein, but you can see that there are lots of arrows right here. So these are the points where the controls are taken place. So when we have the DNA to RNA, there is transcriptional control. When there is RNA to protein, there is a translational control. And these controls are actually, many of these is written in the DNA that we used to think as junk. But nowadays, we got the, we can explore it, and we got the treasure that these are the things that are regulating our difference. So we know the DNA, but many years ago, maybe we thought about the cell, but this is a real picture. So this is nucleus, and it has the DNA is packed in the chromatin. And when we want to sequence the DNA, we need to reveal it from the chromatin structure and from the cell structure. How many of you are fascinated about the Human Genome Project? Do you know about this? Have you seen, have you read the articles? There are 2 papers. So simultaneously, there was 2 papers published in the same day in Nature and Science. But there are many history behind this. I will not go with this. But there were 2 types of sequencing done during the Human Genome Sequencing Project. So one was the hierarchical sequencing and one was the shotgun sequencing. You might know already about this, right? But in the hierarchical sequencing, the advantage was that we got the back clones and we got the physical map. We could have the track of the chromosomes and the scaffolds. But in the shotgun sequencing, it's just chopping the DNA, chopping the genomic DNA in small fragments, and then it become difficult to assemble this, right? So the main problem was the difficulty was in assembling. But human genome sequencing actually was done by the first generation sequencing and a little bit the second generation. But it is now more advanced, the second generation sequencing. You know that we all know about, at least we heard the name, like the Illumina, the ion torrents. These are the main dominant machines we are using nowadays. Also, there is coming the third generation sequencing, Oxford Nanopore and PacBio. So we will have some basics on mechanism of 2 of this technology, actually 3 of this technology. How many of you know that how was the first generation sequencing was performed? Do all of you have the idea about the first generation sequencing? The Sanger sequencing, right? So there was a interesting thing that before the invent of the laser machine that we are now capillary electrophoresis, we used to do it by hand. So the main mechanism behind this was a deoxyDNTPs, the DDNTPs, which does not have which have a missing oxygen here, right? So there is no oxygen here. So it cannot synthesize the DNA. And when it cannot synthesize anymore, it terminates. That was the easy mechanism. But this is a, this was a revolution, this invention, because it was a dye terminating method. And I will not go to details, but the main mechanism is that whenever they get di-DNTP, the di-deoxy-DNTP, okay, deoxy-DNTP, DDNTP, then it terminates the synthesis of DNA there. So if we go for a PCR, you know about PCR, the polymerase chain reaction, we need a DNA there, then we need the normal primers, that flanking things. The 4 DNA nucleotides, DATP, CTP, and then the template to be sequenced, and the DNA polymerase, and the chain terminating DDATP, DDCTP, etc. So the mechanism is whenever they get a DDoxy, the chain is terminated because it cannot synthesize any more of the DNA. And there was fluorescent label dye with the DDNTPs, and we can see the emission. After this, there is a electrophoresis, the capillary electrophoresis, and then we get from the laser, it's detected, and we get these chromatograms of the color. And from this, we can read that where is A and where is T. So the most, which of these will be the first base in the sequence? The small, the shorter one or the lengthy one, the longer one? So I can see that here is a G, it's a pink one G, and there is a blue one. It's called C. So which one will be in the first of the sequence? Earlier of the sequence, the shorter one. So I think we are on the same page now. And there is a video in the YouTube. It's very easier to understand. I will not go with this, but there is a link. You can see it in your free times. So the main problem in the Sanger sequencing is it cannot sequence about more than cannot sequence well more than 800 or 1000 base pair. So if I need to sequence a gene which is 4000 base pair, what I have to do is sequence it multiple times with multiple primers. So there is an example that one gene and I have about 6 primers on the forward side and the reverse side, 6 primers which can sequence it in easy way. But imagine about the 30,000 or 40,000 genes in a genome that we need to sequence. And if we do it in this way, it may not capture all. So that's why the genomics actually came, and the Human Genome Project was a very challenging project. And it was assumed that we may have the solution for every medical disease, or we have the treatment for every disease. Well, we are on the way to that, but about 30 years ago, what they thought, now we are moving towards that. Main term was that, it was like a disappointment that they found that the repetitive DNA, the slide is taken from the actual genome sequence project paper, and they found that only 1% of our genome is exon, which actually code the proteins, and most of it is the repetitive DNA. And what makes the life of a bioinformatician difficult, or a biologist difficult, that the repetitive DNA can mess with the assembly when we get to sequence it back, join it back. What can be happen that if there is tandem repeat, you know about the tandem repeat, it's the repeat that is, in an array. Like here, the repeat motif is GATTA, and you can see that 3 GATTA. What can happen with the short fragments? During assembly, when we want to join together, one of the fragment can be missing. We can join this one with this one, and the middle, these two sections can be omitted. So this can one problem for the repeats, and another problem can be the genome-wide repeats. That is, if I have a repeat in chromosome 1 and the same repeat in the chromosome 3, then during joining or during assembling, this can be, we can mislead by the joining of these two repeats, and we can omit the middle part. So repeat was a main problem during getting the sequence back together. So this is a normal flow. That what are we doing? The genome, the DNA, we are chopping it, we are making fragments, small fragments, then we are sequencing it. After sequencing, we get the data, the reads, it's called the sequence reads, and then we join it, and the first thing we get, the consensus sequence, is contig after assembling. Then we need to fill up the gaps here, and we can make a scaffold, right? So I think you all are familiarized with this. You are all familiar with the terms, but maybe someone does not know, so I am repeating. So contig scaffold, and then we can make the map the scaffolds for assembling the chromosome level genome. So from the Sanger sequencing, what the advantage is, now we have the technique which is called high throughput. So it can generate lots of DNA, lots of sequence, and massively parallel. So at the same time, it's parallelly sequencing multiple times. So this problem, which was happening with the repeats, imagine that we sequence this section about 100 or 200 times, the same piece of DNA. If we sequence it multiple times, then there is a chance that we will not miss it. I mean, we will not be able to omit it, because the strength of high throughputness or multiple time the sequencing, the same thing can get, it's like a replicate, right? When we take more replicates, we get the more reliable data. So for nowadays, we use Illumina sequencing, ion torrent, and MGI sequencing. It's also similar to Illumina. But what we do, I mean, when we get the fragments of DNA genome, but DNA, so we either with sonication or with mechanical or chemical fragmentation, we get the short fragments. Then we need to make the library. So we need to understand how the library is made. Otherwise, we will not be able to assess the quality of the reads we get. So at first we have, we need, this may vary. I mean, sometimes we need more adapters, but the main thing is for Illumina sequencing, we need a sequencing primer binding site. And we also need a region that will bind to the flow cell of Illumina. And also we need index. So there are 3 types of adapter here, which is a primer binding site, an index, and the flow cell oligo. And this is the main DNA that we are going to sequence. And together this, we will call it a library. So this will help the flow cell oligos, the complementary sequence will help us to attach the DNA with the flow cell, right? So this is the flow cell. It's I am showing the high-seq Illumina high-seq flow cell. It has 8 lanes, and we can attach the DNAs here in the all 8 lane, and then massively parallel all of them, sequence all of them. So what happened that with this, the green and yellow, this is the 5 to 3' and 3 to 5' primers, and the DNA, when we pour into it, it joins. Then it makes something called bridge amplification. So it's, you can see that the forward primer is here and the reverse primer is here, and it will join there and make a bridge. And then with this bridge, they will make clusters. So this will called the clusters of bridge amplification, and the DNA polymerase will act on these, and it will create many more DNA molecule from these clusters. So imagine that all of these cluster amplification can give you lots of information about the same DNA. And these clusters actually are, I mean, like the Sanger sequencing, we also have the dye, the fluorescent dye that will attach to this DNA, and it will emit the light, and we can get the signal from this in the machine. So this is called reversible dye terminator. And can you tell me whether the Sanger one was reversible or irreversible? In Sanger sequencing, we also use a dye terminator, but that was irreversible. But this one is reversible because it will make this place empty. I mean, it will give the place to another DNA to come and amplify. And one by one DNTPs will come with the fluorescent tag when it is joined with the synthesis, and the dye will fluoresce. So this is also called sequencing by synthesis. So while you synthesis the DNA, then you are going to sequence it, get the sequence of it. There are also the nanopore and PacBio, which I was telling that those are 3rd generation sequencing. But in earlier days, we used to sequence with the 454 solid helicose. But nowadays, the PacBio nanopore, these are being popular because actually they can sequence large fragments. So in Illumina, the problem is we can sequence about 100, 150, 300 base pair at highest. Well, I should not say that only 300, because Illumina is now trying to update their technology and have the ability for sequencing more length. But the PacBio, it can sequence longer length of sequence. Also that the nanopore. And in nanopore, the easier thing is that it can directly sequence the RNA. Whatever you see that ion torrent or Illumina, these are actually cannot sequence the actual RNA. We need to make the cDNA or complementary DNA from the RNA and then sequence it. But nanopore can directly sequence the RNA. So ion torrent, I will not go into details for ion torrent, but many of you, mainly who are in the targeted sequencing, may find it useful to use the ion torrent from Thermo Fisher or other, because it is good for targeted genomics, not for the whole genome. I may suggest that it's not good for whole genome. What it does is it is depend on the ion. So when we do PCR, what happens? Hydrogen ion releases, right? So whenever hydrogen ion releases, the pH of the environment got reduced. So that change can be captured by the machine, and it can give a peak. So whenever we put one DNTPs, and it can give signal, and we can know that which DNTPs it is, and then get the sequence. So main mechanism of this is like ion torrent is that. And in the PacBio, we saw the Illumina procedure mainly, but in PacBio, how it can sequence the whole or the longer sequence, right? So they actually use a technology which is called zero mode waveguide, and it's also called single molecule real-time sequencing. So they can literally sequence a single molecule, and for this they need to have a chamber like this, and which is zero mode wet, and whenever it is the polymerase is attached in this section, and whenever the DNA come, it can have the dye and get the emission. So if we look into this, so you can see that there are two bulge here, right? So these will help them to sequence continuously. So with the fragments, we attach this adapter, the loop adapters, and this will, primer will be synthesizing, and the primer extension will take place. And we can get more of these continuous sequencing. So it's not detaching, right? So it's we get the continuous sequencing, and then we can get the subset. So this can help in filling up the gaps. You can remember the gaps I was showing during the scaffold or contigs. So it will help literally so to filling the gaps and makes the longer sequence. So what is the pros and cons? Short read. Illumina is getting a short read. It is cost effective and its accuracy is good, but it has a problem. The read length is the main problem, that we might have problem with repeats, or it's not more than 300 base pair. That's a problem. For long read, the main cons is cost. Cost is high, and the raw read accuracy is not very good. But they are improving it, but it's also on the way. Actually, for PacBio, this is not very bad, the accuracy. For nanopore, I found that sometimes it's not that good yet. But that depends on the sequence. I mean, who handle the sequence, that is also a matter. Okay, so any question from you? I think no. So what's next? We are, we doing the sequence, we are analyzing the DNA. I mean, we are fragmenting the DNA, we're making library. then sequencing it through the machine, and we get the reads, the first Q reads. I will go to the first Q later. So after sequencing, we need to first filter the reads, because the reads can contain adapter. You saw that, right? The contain adapters and other primer sequences, and we need to filter that. And for that, we need to trim that. So after this filtering and trimming, the quality assessment, we need to assemble them. Assembly means that we are joining the little fragments that we sequence together to get the whole picture, to get the whole genome. Then you saw that more than 80% of our genome was repeat. So we need to mask those repeat. Repeat masking means that you just highlight those which is repeat. It stays in the assemble sequence, but you just color that or mask that this is a repeat. Annotating the genes and elements. The next step is annotating or annotation, that which one is gene, which one is UTR, which one is non-coding region or promoter. This is the annotating of gene. And many of the projects that we work, I mean, this is one of the difficult things to correctly annotation, do the annotation. Then we get a draft genome which has the annotation and other elements of the genes and other elements. Then we can do comparative analysis or variant analysis, it's called. So we all are human and we have the similar type of DNA, but as I said that we have variation, right? So targeting those variation is the variant analysis, so that we can target that why I am getting the disease, but you are not getting the disease. Maybe there is something hidden in the DNA, right? And then we can do the expression analysis, the gene expression or the protein expression, or nowadays the metabolic expression, and do the functional genomics to prove that this is the cause of the main disease, or this is the cause that why I am not getting the high yield in a rice or other plant. So these are the basic steps that we need to go through. So if you want to initiate a genome sequencing project, or you say that you want to do the genome sequencing of tulsi pata, tulsi leaf, then maybe you have to design your experiment accordingly, that what are you trying to get from, I mean, what information you are trying to get from this, that why do you want to sequence the tulsi leaf? So genome looks like this, the gibberish A, T, and CG. But when we annotate, we can see that in which part we have the exon, we have the 5' UTR, 3' UTR, and where is the promoter, and where is the intron, and the intergenic region. This is very important to correctly annotate the genome, because if we want to reveal something new or identify something novel in our genome, then we actually do the annotation by the existing reference genome or other available gene sequences. That's called the reference annotation. But there is another one which is called ab initio, or we just see the structure or the motif hidden in that sequence and then annotate this as UTR. For example, you might know that there is a sequence for a splice junction, right? Also, there is a sequence for promoter. So most promoter will have the Tata box, TATA. So these are called motif. And our genes mostly start with the ATG, this sequence. So these can be the identification motif for the program to explain or to identify this as a gene or intergenic region. And we all use the genome browsers. So genome browsers are a very good way to show us, I mean, the genome is very long, but we can, in a browser length, short way, smaller way, we can see the whole genome together. So from this discussion, we now have the idea that main three types can be of the genomics: the structural, functional, and comparative. So comparative is the one that I was talking about, that it compares the gene from different genomes. So you maybe heard about the 1,000 Genome Project. Also, there are many more about for other species. Like there are 3,000 Genome Project for rice plant and 10,000 for the other plants. Functional genomics, it refers to the study of functioning of gene and their regulation and product. So the gene expression, when we do the RNA-seq, we are going to target the functional genomics part. And the structural, it deals with the determination of the complete sequence of the genes and gene map. So mainly the DNA sequence and the variation in, not the variation, the DNA sequence annotation. This is dealing with the structural. So I have got this workflow from a textbook. So at first we need to do the laboratory work, which involve the experiment design, library preparation, enrichment analysis or enrichment. I must say about the enrichment, because when we say I'm preparing a 500 base pair library, so we have to target those 500 base pair PCR products only. So that's a kind of enrichment. So when doing when preparing the DNA for genome sequencing, we actually do the PCR in most of the cases. So so that's a targeting the specific regions. It's called the enrichment. Then in the next generation sequencing, we can use any of the platforms, and we get the output as FASTQ. So FASTQ actually of two types, as Sanger and Illumina, but we will not go into that details now. But mostly what we get from the sequencing facility is the FASTQ reads. Then we have to jump into the analysis pipeline, which is the quality assessment. For trimming, filtering software, we will use the FastQC. Maybe many of you have already used that, but there are some details that you need to concentrate. Then we need to align the sequence, the filtered reads to the reference genome. And we can do that using BWA, bowtie2. And the output will be in a SAM or BAM format. SAM and BAM both are alignment mapping format, and BAM is the binary, and you cannot read that actually with your eye, but computer can, and SAM is the thing that you can read. Then we go for, if we want to do the comparative analysis, we go for the variant identification. And for that, we can use GATK, so Genome Analysis Toolkit, or SAM tool, realignment, recalibration, and the output will be the VCF file. It's the variant, it's a list of variants. I mean, the VCF stands for variant calling format file, and it's a list of variants. Variants mean like SNP. We know about the SNP, right? Or insertion or deletion. These are all variants. And it's a list of variants. And where are they? I mean, are they in the gene or in a UTR region or in a promoter region? That's the variant VCF file. Well, actually, the original VCF file that does not say that in which region it is. We need to annotate the VCF file to get the idea that where it is actually residing. So this next step is annotation, comparison to public database. For example, DBSNP, then 1000 genome functional consequence score, and that can give you that actually where is the variant is locating. Then after we analyze analyze it, we get a visualization. We need to visualize it DNA, I mean the sequence we got, the VCF file can be visualized, the BAM file, or it can be also visualized. The BAM file will show you that which reads are, aligned to which part. Then prioritization, discovery of relevant variant software, for example, polyfen 2 VEP. These actually prioritize that we get lots of, based on the filter we set, the threshold we set, we get lots of VCF, lots of variants in the VCF file list. But we need to prioritize that which can be a functional variant. By functional variant, I am saying that which can alter the protein function and which can alter the gene expression, etc. And finally, the storage. Storage is nowadays a big problem also. But we need whatever we are sequencing, we need to store it and dump it to the public databases so that other can avail. That's a good practice, that other can avail and work on that data. Because when you are publishing the results already, you may not be interested or you may interested in a specific site, but other may find it useful and they can be interested in other side of the sequences. Okay, so any question from this part? So, are we on time? I just wanted to, okay. So maybe I will go with the details of the files. So this is two single files, that is FASTQ. And do you know the meaning of the things that we can see here? How many of you do not know, or how many of you know? Okay. So this is the file that we get, the FASTQ reads. You can see that at the rate sign right here, at the rate HWIST911 something. This is actually the instrument name. So it has 4 lines. The first line starts with this at the rate sign. Then there is a sequence, ATGC, then a plus, and then this is called the quality. These are in the ASCII character, and the computer can read this as the quality of the base, that how how reliable the base is. That here I have the A, how reliable it is. It's the indication of this is the quality score. And in the header, this is called the FASTQ header. We might know, everyone might know that the FASTA header, FASTA, that the normal file that starts with the greater than sign, that's the FASTA file. And this one, the FASTQ have in the header, the run ID, the instrument name, then the 111, this is the run ID, then the flow cell ID. The flow cell refers to the Illumina flow cell here. So, and it's the 5th lane, flow cell lane. There, I saw that I told you that there are 8 lanes in the high-sec Illumina. So why this is important? Because when we will do we might not do it here, but when you will do the RNA-seq analysis, you will say that you see that you need to do the normalization, because in lane to lane, the sequence output may vary. So this may vary lane to lane, and that's why we need to know that in which lane this sequence was performed, so that later we can normalize the sequences based on the lanes also. Then 1101 is the tile number within the flow cell. The coordinates the member of a pair. Okay, this is important. One is here, right? This one. You can see 1, N, and 0. 1 means that this is the forward read. We can have 2 types. If we are going sequence for paired in read, then number 1 is the first pair and number 2 is the second pair. And then we see that N, Y, it can be Y also. Y, if the read is filtered, N it is not otherwise. So after we filter the sequence, we will filter the FASTQ sequence, then you might observe that whether it is still N or Y there. And the latter one, these one are the index sequence that we put with the library. So the 18, this one mostly here is 0 when none of the control bits are on. So in high 6x system, control specification is not performed and this number is always 0. But if there is control specification, then this number may vary. 18 or 23, it can vary. So why it's important for us to understand the FASTQ file? Because if we start with a, not so good file, then we might get a misleading information about the genome. So two things came here, the paired end sequencing, right? There are two types of sequencing read, the single end and paired end. When we talk about the paired end sequencing, we actually sequence it from both ends, but why it is important? It's not like that this is the 100 base pair, say 100 base pair Illumina read, and I'm getting the sequence from forward from this side and reverse from this side. Not like that. Think about it as a library. Say you have a 500 base pair library, and when you are sequencing, you can get 150 from this side, also 150 from this side. But when you got both the read, you know that, you will definitely know that these two reads are 200 base pair apart. Since these are 500 base pair library, and you are sequencing from one side the 150, the other side the 150. So they must be 200 base pair apart. And this is important because previously in earlier days, we needed to make a physical map by genetic mapping or by linkage mapping. We needed to construct a physical map or genetic map for the before doing the genome sequencing. But this can help this indication that, this is kind of a helping in scaffolding. I can have the sequence from the single end, but with the paired end, my genome sequence will be more reliable, but it will be more costlier. But as you know that paired end sequence means I'm sequencing from both sides, so it will cost double. And there was another term you might heard, that mate pair sequencing, which is not much used nowadays. But in mate pair sequencing, a similar kind of thing was happened. Like there was a linker, the yellow one you can see, and it can make it circular, the DNA. And then we chop it with a restriction enzyme, and then we sequence it. And also this is this was also used for tracking that how apart the both reads are, because since the linker is known, the yellow part, it is known to us, so we know that how much distance they have between. So both of these help actually to get the reliable sequence or making the assembly more reliable. So in Illumina, the quality score, the range is from -5 to 62 in Solexa, that we are not maybe using. In Illumina, it's 0 to 62, and in Sanger, it can be 0 to 93. So what does this quality score actually mean? If I say that my base pair quality score is 60, it means that 1 in 1, then 60, 1 in 1 in 60. So it's what 1 million, 1 in 1 million error rate is allowed. So probability of incorrect base call. So if my quality score is high, then my accuracy is very high, like 99.999%. So you have to think in this way, that quality score 10 is much bad, or it's not very good. Consider when a quality score of 60. And the quality line that we saw is the ASCII character. You can ASCII code, you can find it anywhere, but you can match to get the actual quality score from here, from this list. Well, you will not need to do that because software will do that for you. But if you need to analyze and make a decision on some specific spot, then you can do that. Okay. So any question from the FASTQ file description? No. Assessing the data quality. So whenever we get the reads in hand, we need to jump in for the data quality assessment, and we normally do that with FastQC. FastQC is a nice software. You can easily install it. I will show in the hands-on portion. But what does it say? It's a very good par-based quality graph on the left-hand side. You can see that these are the box plot. You know about the box plot, right? It shows the distribution of the data. And this x-axis is actually not on a scale. You can see that in the beginning, like from 1 to 9, it's the bin for 1. Each base has 1, but in the middle, it has a frequency of, it's a range. And again, in the later portion, there is a individual bin. But what it tells that these in these bases, it's a sequence length is 150, and in number 3 position, the quality score varied from 32 to 37 maybe. So it's not a very, it's okay, but it's not a very high quality score. But in the middle of the section, 45 to 49 number position, the variation was not that much, and it was near 32 to 40. So this tells us at a glance that whether my data is actually good or bad. So it's a bad quality score, and you can see that the range is from 0 to 39. There's a very much variation. So when we want to filter this sequence, we actually will trim this from here, because I think this is a very bad sequence here, and it's from 165. So I will tell the filtering software to cut it from 165 to the end. So normally it will lose. It will. The disadvantages, it will lose some of the data, but whatever I get is a reliable one. And since we are doing the high throughput parallel sequencing, so for one position, we have lots of data. So in that case, that will not be a problem. Per sequence GC content, the ideal GC content would be the blue one, the normal distribution, but this one is actually a good score, because it has a GC content distribution near to the ideal one. So there a good per sequence quality graph. So in per sequence quality graph can be assessed also, and we can see that here how the bad sequence will look. Adapter content. We are putting the adapters during library preparation, right? But in the machine, which is doing the sequence and generating the FASTQ rates, that actually have a trimming option that omit the adapters and the other sequencing primers sequence from our reads. But there might be some adapter remaining, and we need to assess and cut those adapter or filter this before proceeding to do the assembly. Also, how many of you know the concept of K-mar? Any mar. So K can be any number. And here we can see that these sequences are actually overrepresented. Like TCG, GAAG, this particular sequence is a red one, and this is overrepresented in the first end of the sequence. Also, in the first end, there are multiple K-mars that we can see that overrepresentation. And this says that the first part of the sequence is not very good. Also, the N means any nucleotide, or when the sequencer is not sure about what nucleotide or what base it is, then it puts N. So per base N content can be also a good indication that whether this is a good or bad. This is a bad sequence because it has N in the middle. Okay, so when we will do the first QC hands-on, we will see it more on, we'll understand it more, I think. Then we need to go for the trim and filtering the short reads. Trimming and cutting or masking the sequence from low quality score region, beginning or end of the sequence major target. Then removing the adapters and filtering with low mean quality score. Say I can give a threshold of 30 or 35, that below the whatever reads there are with below 30 quality score, we can omit that. With too many ambiguous N bases, we can omit those sequences, or there can be some sequence that is only 50 base pair length. We can, and the other is 150, then we can omit the 51, the two short ones. So for trimming and filtering, what we use, the tools we use is varying because time to time, because in earlier days, I used to use the PrintSec or the, no, sorry, not the PrintSec, the cut adapt. This one was simple and fast, but then we use the ultra configurable Trimomatic. But what I found nowadays is being used is fast P, and it has the simple and ultra configurable both option available. So fast P is a good choice maybe. So it filter or trim the data according to quality. We just need to input the actual reads, the raw reads, and then get the output as the filtered reads. So any question? Assembly. So next, our next step will be to assemble the genome, assemble the reads to get the most of it, the sequence. Assembly can be of two types, the de novo assembly and the reference guided. So de novo means something new. So from the scratch, we can build up the sequences and get the assembled genome. But for the short reads, this can be a problem. As I said, that those are short reads and repeat can hamper the assembly process. And most of for most of the organism, you will see available the paper published. These are the initial assembly or the draft assembly. And then they do the scaffolding and finalize it. So you might know that the human genome, the finalized version of the human genome is just came out a few days ago, right? But it was published during 2000. So it took about more than 20 years to finalize it. So in de novo assembly, what we do is you all used to with the puzzle set in your childhood, or maybe nowadays there are puzzle set for adults that has more than 10,000 pieces of puzzle. Imagine that making those, reassemble those puzzle set, and it will have this kind of scenario. But in what it does, that it has two type of algorithm actually. One is called the overlap layout consensus, or OLC, and the other is the de Bruijn graph. So both have advantage and disadvantage, but the de Bruijn graph one has the kind of mechanism. The OLC has a different kind. So what OLC does is like this: the overlaps are identified and they are matched, and make the consensus sequence is identified from those matching paths. And in the de Bruijn graph, they make a KMR, or they try to assemble it, for example, 21. So they will take 21 base pair and then another 21 base pair, fragment the whole sequence in 21 base pair, and then try to assemble those 21 or 31 or 127. So the more the k-mer is, the 127 mer will take longer time because it's difficult for matching it 127, but this will be more reliable. Yeah, because the longer the k-mer, the more reliable the assembly will be. But it will take time. This method was designed for short read technologies, but here also repeats are highly problematic. Overlap layout is slow and time consuming. Deep routine is fast and loose. You might be familiar with the name that Celera, Canu, HGAP, Falcon. These are the this follows the OLC, and the SOAP de novo, Abyss, Allpath, Velvet. These are some name of the tools that we can use. This follows the deep routine graph. So in our hands-on, we will use the Ray, which is a mixed assembly software that combines the two methods that use both the method, and it actually is more, I mean, it's better to use the mixed assembly software. Original reads are not discarded and instead mapped to the contigs to generate the scaffold. So when the software actually do the assembly process, it can be like more realistic graph will look like this, but we may not need to go through this because this is more inhuman for us. So what ruin the graph? I mean, this is the ideal graph that you are seeing, that they will find the overlap and connect them to build the consensus sequence, but there can be read errors. Introduce false edge and nodes, non-haploid organism. Heterozygosity can cause us lots of detours. So we sometimes work with a plant that has 6, I mean, that is tetraploid or hexaploid sometimes. That makes life very difficult because they have, for one position, they have multiple bases inbuilt. There can be repeats if they're longer than the read length. If the repeats, keep it in mind that if the repeat is longer than the read length, so your read length is 100 base pair or 150 base pair, but the repeat is like 200 base pair, then it will hamper. I mean, it will hamper the assembly process. Causes nodes to be shared, locality confusion. So where the read actually is in the genome, that can be a confusion due to the repeats. So we know that there are different kinds of repeat. I will not go into detail for this, but we know about the satellites, which are the tandem repeats, mini satellites, also microsatellites. Microsatellites are a good marker also for doing the genotyping or linkage mapping. Also, there are genome-wide or dispersed repeats like the retrotransposon or the DNA transposons. And these are the main repeat content in our DNA sequence. There are more extensive classification of these, but these are the culprits which are making our assembly difficult. So here, that I already stated this too. The law of repeats. I found it in another slide, that it is impossible to resolve repeat of length S unless you have the reads longer than S. So I hope you can understand this, that if you have a repeat of more than your length of the reads, then it is very difficult to resolve. Contig sizes are limited by the length of the repeat in your genome. You can change this, and the length of the reads, if you use the long read technology, that's why the PacBio is very popular nowadays. If you use the long read technology, it can make things good. So single end and paired end, I already talked about this. Paired end is actually better for the scaffolding purpose. And this is the scaffolding things that filling up the gap, right? These are the contigs and you can fill up the gaps to build the scaffold. And then after building the contigs and the scaffold, there is a assessment of the assembly, assessing the assembly, how good it is. We desire the total length similar to genome size. So say we know that our human genome is 3.2 GB. Then after assembling the genome, we might desire that it will be the 3.2 GB file. Fewer larger contigs. So contigs, number of contigs, if I have larger contigs and fewer, not very much, not many, then it will be a good one and the correct contigs. So for this, we use some metrics like N50. Have you heard the name N50? So N50 is the one of the metrics that can give us the idea how good is our assembly. And there is also a thing that coverage. I will talk about this later also, that we can calculate the actual coverage of the genome. So what is N50? Is the length of that contig from which 50% of the bases are in it and shorter contigs. So say you have 100 or 1,000 of contigs, and then here, imagine you have the 7 contigs. You need to sort them first, right? So these are in order, and then maybe you can calculate the total of them. The total summation is 50. Since this is only 7 contigs, this is easier for us to calculate, but for the 1,000 of contigs, we need software. And you can all easily write script for calculating this type of things, the assembly statistics. So the total is 50, and now N50 is the halfway sum. So 50, half of 50 is 25. So the sum, when we get the greater than 25, is the main point of our N50. So here we can see that 1 plus 1 is 2, then 3, 5, 5, 10, 8, 18, plus 12 is 30. So when we come to number 12, then we get a summation of 30, which is greater than 25. Right? So that's why it is called the N50 of this assembly is 12. Because N50 is the summation, halfway summation of the N50 means 50th. Means 50th. So this is the halfway summation of the total ordered contig length. So if we have a N50 of 3, will that be better, or that's not good? Which one is good? N50 of 3 or N50 of 12? N50 of 12 is better because it is saying that most of my contigs in the assembly file has the length 12. So it's a greater length. The more shorter the length of my contig, it's bad. So if I have a N50 of like 20, that's N50 number is 20, that is much better. So higher it is, it's good. And another is the coverage. There is a law. I mean, there is an equation, C equal to ln by G. C stands for the coverage. G is the haploid genome length. L is the read length. And N is the number of reads. So L is the read length, like 150 or 100. G is the haploid genome length. So maybe it's, for rice, maybe it's 434. And N is the number of read that I got. And from this, we can calculate the coverage. But in general term, the coverage means if I say that I have a genome assembly of 50x coverage, that means that for each base of the genome, I have at least 50 reads. I mean, at least 50 times that base was sequenced. So when I deal with a 14x genome and when I deal with a 50x coverage genome, 50x1 is much better because it's more reliable, because it's at least one base is, each of the base is at least sequenced 50 times. But that causes the cost high. Okay. So, okay. I was talking about the draft assembly and the closed or finished assembly. And you might get the idea that which one we actually say as the draft. The closed one is actually, we get a good annotation, takes a lot of work. We also need to validate some through the functional genomics, and that's a closed or finished assembly. Many of you might want to do the metagenomics because some of you work with the environmental science and maybe some work with the gut microbiomes. And metagenomics for bacteria and eukaryotes, bacteriovirus or eukaryotes, we have separate set of software that we normally use. So for the eukaryotic genome, we can use these softwares, or these tools. But for the metagenome, we might use the MetaVelvet because it has the specific algorithms that can do the meta-analysis for the metagenomes, or we can use the custom script. There are specific tools for bacterial genome also. For transcriptome, what is the transcriptome? The RNA content of the, not the genome, the transcriptome is the RNA content of our body, right? So it is dynamic, right? Because when you are in stress or when you have a disease, it can change. The transcriptome content can be changed, and the transcriptome content of a cancer cell and the transcriptome content of a normal human may not be the same. So when there is more dynamism in the transcriptome assembly and sequences, and we need to do the trinity. And the main helpful thing is there are not much repeats. So transcriptome assembly is better, I mean easier, because they don't need to deal with the lots of repeats. Okay, so we are kind of at the end of this session, I mean this lecture. We can continue the other things if we have time. Okay, just wanted to touch this one because this is a very basic thing that for when we did the mouse genome assembly or the earlier one, the Drosophila, the human one, we needed a physical map to correctly track the chromosomes, 1 or 2 or 3, but which comes after which. So for this, we needed the physical map, and there are many tools, many wet lab tools that we need to do for getting the physical map. And for genetic map, we need the pedigree information or the breeding information, cross-breeding information, and we can construct the genetic map. But you can see here that not always the location are same. For example, this gene is actually in the here in the physical map, and both of these can help finalizing our assembly or the sequence map, and that can be a good resource. If we have the physical map, that is a very good, I mean, we can desire for a very good assembly. So we were talking about the de novo assembly, that constructing the genome from the raw reads, but we can also do the reference assembly because for 1,000 genomes, that we have already we have the reference human genome, right? But that is actually another model. I mean, that cannot represent all of us because when it was constructed, it was taken from different people, but not it was not covering the all region of the world. And for one of the private company, it was the DNA of the owners that you might know. And this was alignment map. Oh, by the way, we can have our genome sequence right now. You know about the 23andMe, right? I mean, there are some this kind of this kind of company that can sequence your genome or a specific ancestry or a specific disease information for you in less than $1,000. Sorry, and the whole genome less than $1,000, and the information only you can get in $100. So for reference genome, we need to have for reference mapping, we need to have a reference genome. And you can see here that the reads are actually mapped in those genome, and it's easier, right? Because instead of constructing them from the scratch, if we have a reference and we can just patch it with, align with it with this, it's easier. Map to best region, how to handle the multi-map reads. So say one read is mapping in the 3 region. Then how we take the decision that which one we will take. So we need to see that which is mapping the best region, and which one is randomly mapping, and then we can have a alignment score for this. Say for all of this, we can have an alignment score, and we can keep the better alignment score and discard the others. So we already talked about the paired end sequence. And for reference genome, it is also helpful because in the reference genome also the more sequence, the basic thing is that more we sequence, the more reliable this will be. And if there are insertion or deletion, that I might share a story that there was a rice genome it was published that has a 200 base pair deletion in that gene, and which encodes for potassium transporter, phosphate transporter, and that was not present in the reference genome. So this kind of discovery can happen also with the reference genome. And for de novo assembly, for reference genome, you might escape this, that because in the reference this is not there, but in de novo assembly you might find this 200 base pair chunk which was not present in the reference. So that's why we need to do the de novo assembly also for newer species, and also for the older one which already been sequenced. This is called a resequencing projects. And also now there is a concept of pan genome. You might also consider this, because pan genome analysis sometimes can give you the information that is not available in the reference one. And also there, if we have more than one reference genome for one species, that can be a good idea to get more reliable information. Okay, so mapping tools, we can have lots of mapping tools like RNA star. RNA star is for transcriptome mapping. BWA MEM2. This can be a Burrows-Wheeler alignment of software. And this is mostly being used. And another one being used is Bowtie, which is not stated here, but these are the mapping tools that we can use. And all of them have advantages and disadvantages also. So shall I proceed, or I should keep it for after the hands-on session, because maybe there are lots of information we are dumping. Okay, so I will go with this in the hands-on session. So any question for now? For the mapping or the assembly or the whole workflow? And we can also talk about the experiment design. For genomic sequence, there is not many things to consider, but for RNA sequencing, we need to carefully design our experiment so that we get the correct information. So if you do not have any question right now, we can stop here. All sort of thing? All sort of covered today. Oh, Well, that will be difficult for this session, but we will do the one of assembly. We will start with the filtering, then trimming, then assembly, and later we will do some mapping, the alignment. Yeah. Yeah, variant analysis can be also predicted if we have time today. Otherwise, we can take it, we can see it the next day also, because after assembly, if we have time today, I have the scripts here, we can do the variant analysis also. Yeah. But for RNA-seq, we might not have the time today, I think. Yeah. Okay. So repeat masking mean masking the repeats. So you identify that this is a repeat and color it with a gray thing. I mean, if you see in NCBI genome or any genome sequence, you might see sometimes that there are base pair with small letters like A or C. So these are actually masked or they are dimmed because they are, they are simple sequence. They are not complex sequence. So when I talk about a repeat, it's a simple sequence, right? Only A or AT like this. So this is a simple sequence, not a complex one. So whenever the repeat masker that find this kind of simple sequence, they mask it. And they consider it repeat. Also, there is repeat DB, repeat database already available. So they actually align the assembly with this repeat database and then also mask this kind of sequences. So repeat masking is a very needful step in the assembly, genome assembly. Okay, so shall we stop here now? Okay. Okay, thank you, everyone. We will proceed from this step later. I forgot to ask, any question from the the audience in Zoom? I think there is no. Okay. Okay. Thank you. Okay, thank you, everyone. We will proceed later. Yeah. আমরা কি টাইমের আগে চলে আসছি নাকি নাকি টাইমের পার হয়ে গেছে 10:30? Okay. Well, I might start the hands-on. What do you say? আমরা শুরু করে দেই, রাইট? আমি যে সুন্দর করে সবার কাছে বাইটাই নিয়ে নিলাম, এখন কি আবার শুরু করবো কিনা এটা চিন্তা করছি। সবার কি কোনো কনসেপ্টে কোনো ক্লিয়ারনেস দরকার আছে কারো? Sure. So we are back. চলে গেছে বোধহয় না? না, যায়নি। Okay, so let's start the hands-on session. How many of you do not have the Ubuntu installed in your own laptop? Ubuntu or any kind of Linux installed? Okay, so we will do some exercises in our own laptop now, and then in the later cases, we will go to the HPC. So at first, we need to see whether we have the FastQC installed. So can you just check whether it is installed? Just type that FastQC. And consistent বার করবো? এই ফন্টটা কোথায়? ও আচ্ছা, এখানে আছে। হলো না? আচ্ছা, আরো বার করবো? কন্ট্রোল প্লাস অসুবিধা নাই, আমি এটাকে আরেকটু বড় করে এখান থেকে বড় করি। কন্ট্রোল দেখেন তো মানে হচ্ছে না কিনা। বেশি বড় হয়ে গেছে। ঠিক আছে, এখন ঠিক আছে। Okay, you can just check whether the FastQC is there. So you can write FAS and then put the tab, then Q, then tab. So you must be familiar with the tab option because it makes life easier. You do not write or type the whole thing. So if you have it installed, then it will show up. And you can have this help options for this. So we all are in same page. We all have the FastQC installed, right? And then we will need to download our reads. I have collected some reads, which is in the Slack I uploaded. So I hope you all can access the Slack channel course materials, and there you can see the SR, the data starting with SR, that's the reads. And there is another thing, the GCF, that's our reference genome. So we need to download all of these 3 files. Yeah. And also last night, Amitabh shared notes with you that there is the, what does it call? The notes, the commands. So it's like this. We will go one by one. So quality analysis and filtering. For this one, we will just write FastQC, and we need to go it in organized way. So in bioinformatics, the main problem is we have lots of data, and sometimes we get the disorganized and we cannot find the actual data. So at first, make directory, mkdir, and make a directory for FastQC or FastQ analysis. Okay, and then you can download the or shift your sequences here. Why does I have this? আমার কাছে কোথায় আছে ভুলে গেছি। Okay, so you all have the genome, the reference, not the reference, the FASTQ file here, right? And you can see it by typing ls. Okay, so you can say, you can type, you can go to the directory you make, which is FASTQ something, and then you can type ls to list all the files there, right? Now we need to analyze these or quality assess these two files. You can see that one of the name is R1, which is a read one, and the R2, the read two. So these are the paired in read. Let's check it first with how does it look. So just type LESS, which is less, which is not LS, right? Less. I mean, I am seeing this in a less visit. So less than SR, then tab. You do not need to type all, then tab. Then you can just put 1 and then again press tab. So I will see the in limited version of this sequence. And this is how it looks. So it has, as I said, that there is a 4 lines. So it has @ it starts with the @ sign. That's a header, then the sequence, then another line containing the plus, and the last line is the quality file. So quality of each bases. So for this is one read. Then this is another sequence, and you can go down to see the other. If we want to see how many sequences are there, we can have a trick. Are you familiar with this command, which is called grep? It can extract information from a file. We can see grep and then -c. -c stands for count. That we are going to count the number of @ sign in this file. Like this, grep space under -c for count. Then what are we going to find? That's in the quotation. And then in which file? That's in the last portion. We will press enter. And it says us that there is a 140800 @ sign. Also, if we want to see if the paired end file have the same number of read, we will do it by, we will just press the upper arrow and then shift it to R2. It's not the same because there might be some errors. But in the paired end reads, normally we need to have the similar number of sequences. Anyway, that is actually not the main quality assessment. I showed it to get you used to with the environment of the Unix. I hope we all are in the same page now. Okay, we download Have you downloaded it from Slack, the Slack channel? Okay. So where is it now? It's in the download folder, I guess. But in your home, can you make a directory which is named as the first Q analysis, or you can name it anything, but make a directory with the mkdir, this command. The command is mkdir, like this one. And I prefer to keep it in a separate directory so that I do not lose the data or do not lose the track of the data, that where it is, because there are many files in the computer. So after making the directory or the folder, you transfer or your downloaded data there, or you can directly download your data in that directory. And whether it is correctly downloaded, you can see it using the ls option, which is list all files in the directory. And after putting the ls option, I found that I have 2 sequences in that directory, 2 files. And the folder I named is first Q analysis, but you do not have that one. You have to make that one, because I uploaded the files, the read first Q files, in the Slack channel, Slack class course material channel. It's just uploaded recently. You can find it with SRR something. Data for downloading is the post name. Okay, so after downloading anything, it's a good practice that we can, if we see that what we have in the file. So that's why I put the less option, I mean the less command, the LESS, and wanted to see how does the data look. Okay. And then I wanted to see that how many sequences are there. So I use this grep option, the grep command with the C option. C stands for count. And I tried to find how many @ sign is there. Oh, you need to put Q. If you use the less command to enter into, to view the data or view the file, you can just press Q to exit from that interface. Okay, so there are variation of less. You already know maybe that that was shared, that we can see only the head portion or only the tail portion of that file also. For example, head stands for the upper portion of that file only. It will not show for this. Because it was not showing because it was in the Ganji formation. So grep command just fetch specific sequences from your file, and it will fetch all the lines that has that @ sign. And when I put the -C option, it will count that how many of this @ sign is there. But notice that this one is in GunZip. You know about the GunZip, right? It's the archive, or the zip one. It's a super zip, so GunZip, GUN. But we do not need to unzip, or we do not need to elaborate this file because it's easier to transfer. It is in GZ format. I mean, the GZ format is easier to transfer or transport. So we will not unzip it. We will use this as it is. Okay, next we will go to first QC analysis, and we will put first QC, then SR something, the first one. And if we enter after it, it will create the analysis. But we can do another thing. We do not want to write all the name individually. we can just put it like this and then press a asterisk sign. Asterisk is a wildcard type of sign, and it can analyze all the file that have this portion. I mean, this is a common. Why this is common? Let's see again that this one. Okay, গ্রেপের পর এখন আমরা হচ্ছে ফার্স্ট কিউসিতে we will start with the first QC. So let's see that we have these two sequences in the folder, right? We have both of these sequence in the folder, and they have this thing common, that SRR something 500K R. They are different from here. Here it is 1 and then 2. Otherwise, for the previous portion, these are the same, right? So we can do something easier here, that we can do first QC, then SR, then put a asterisk to get all analysis together, to get the all analysis done together. If we had 3 sequences, then it would make all of the 3 sequences together. So at first they analyze the R1, then they're doing the R2. But we do not need to do it by this asterisk sign. We can do it other individually also. Like the first QC, the command is first QC, then a space, then the file name. Like this. And it will also do the analysis the same way. All of you could do till this step. So these are the very basic step. Actually, we are now having both of the, for both of the file, we are now having the quality assessment. And you can see by writing ls, by typing ls, you can see that in the folder, now we have 6 files instead of 2. Instead of 2, for each read, there are 3 files. So one is the zip format, the GZ. Another is the first QC analysis. And there is an HTML file also, and a zip file. So if we now go to the Ubuntu GUI, or the folder here, this is the directory in my home, which was named first Q analysis, and just locate the HTML file you got. And if we double click this, we can see the report of the first QC analysis. And these are individual report, and we can see the green mark stands for it's passed and the cross is not passed. So for this specific data, which is the R1, read 1, we have this number of sequence, the length, and % of GC is stated. The par base sequence quality. Is it a good one or bad one? It's not very good, but it could pass their threshold. So when it can pass their threshold, it can be considered. Maybe if we trim from the lower the end, it will be better. Then we have the part sequence quality score, which is also passed and not very bad. Then par base the sequence content. This is bad actually, because it needs to be horizontal, and it needs to be more similar to each other. I mean, more parallel, but here we can see that there are more jumping and it's not very good. So we need to trim this side also. Per sequence, GC content is not very good also because the red one is not, there is some difference than the blue one. Per base N content, it's very good because there is no end content. Sequence length distribution is also good. Overrepresented sequence. Overrepresented sequence, it could not pass because there are some sequences, some adapter sequences here, we can see. Also, not only the adapter, there can be some specific sequences that are overrepresented in this side. We can just omit or trim those. And adapter content, there is not adapter content, and it was passed. And we can also do the same thing for our R2, which is the read 2. And we can see that per base sequence quality of read 2 was very bad. You can see that here from the box plot, we can see its distribution is very bad. So it could not pass. Per base sequence content is also bad. And there is overrepresented sequences. And here you can see that which sequence is overrepresented, and you can track that whether it is adapter or any primer or something else. So here it is this particular sequence came 712 times in the read. So it is not a very good indication, and thus you can get the first QC report. After this, you decide that how much length you will keep, or whether you will, what threshold you will decide, like maybe you can keep only the quality score below upper than 30 or 35, something like that. Okay, so for this part, what we have done is we downloaded the data, our reads, and then we analyzed this with the first QC tool, and which gave us a graphical representation of the data, that how much good is our data. And we found that the R1 is more or less good, but the R2 needs some trimming or something. Okay, so are we all done with this? Then we can proceed for the filtering or trimming, unless we have the call for tea. 10:39. Okay, so for filtering, okay, so in my notes earlier that we were actually using, I mean, I was planning to use the HPC, the AWENS supercomputer for accessing the data, but it will need the password. So we did not do it in this way. We use the Slack channel. So we are starting from here now. Okay, now we will use the first P tool. First P, it will be used for trimming. Other tools can be used for trimming also, but we found it easier. So just check whether you have the first P option installed. If it is installed, and then you will get the manual page or the help page like this. And if it is not installed, then it will ask you to install the first P. To install first P, you can just type sudo supercomputer super user do, then sub apt from the then install, then space first P. Try this one, sudo apt install first P. It will install the first P tool from your repository. From Ubuntu's repository, it will install it. In my computer, it is already installed, so I am not going to install it again. But you can try this command to install this. And then You can just use this command. We will go through it step by step. So the first part is first P. First P is the command. Okay, is first P installed in everyone's computer? Okay, try writing the command first P, that is the main command, then the options. In the manual or help section, you can get the explanation for each option. But what it is saying that detect adapter for so adapter ache kina, it will find whether there is any adapter sequence. Over-representation analysis, it will also analyze for over-representation, and then if there is, it will omit those over-representative sequences. Cut right thread 2. Number of thread in our computer. We will use 2 thread. In supercomputer, we can use more than 1 thread or 2 thread. It depends on the structure of your computer. HTML, if we want to generate an HTML output from the analysis, and this can be named as trimmed first p.html. So this part is for the analysis output name. We can also output as a JSON file, but it's not mandatory. But these two, both are HTML and JSON. We can name these as trimmed first p or trimmed first p HTML and trimmed first p JSON. Then minus dash I means the input file. The input files are these. The first one R1, and notice that the second one is uppercase I. The second one for the paired in sequence, it's uppercase I, and it the R2 one. So smaller case I and then uppercase I, then dash O. O is the actual output file that is already filtered. So the filtered file, we will name it as trimmed. I mean, we just added a prefix that is trimmed before this SR and the whole name. Then again dash O, which is in the uppercase for the R2. And this is trimmed something R2 first Q GZ. So can you try writing this instead of copying? So first try writing first P, then a space, then detect adapter for PE. Orthat, I am telling it that I'm providing a paired end sequence and asking it to detect the adapter for PE. Space, detect adapter for PE. Then I am asking it to analyze if there is any over-representative sequence. So just a space and again write dash dash over-representation analysis. Then I am asking it to make correction for this over-representation. Correction. Then I am asking it to cut right. Kondike cut, in which portion it will cut. then I am asking it to use 2-thread. In your computer, it might not be needed, but for submitting in HPC, we will need to provide that the number of thread I want to use. So if we use more threads, then it can be faster sometimes. And then I am asking to provide the HTML and JSON files. This will be the output file in HTML format and JSON format. Then just notice the input format, that minus small case I, which is our main R1. Dash I টা হচ্ছে আমার input file. That's an input file. So our input file is a raw read first queue. So minus I holo is R1 dot first queue. Then uppercase I is the second pair. So it will be R2 like this. Then the output file, which I will name as streamed. And the uppercase output file as 2. Okay, so this will be the command for first P, and we can just press enter and see how it goes. It will take a bit time because it is generating all the sequences again. Detecting adapter sequence for read 1. So you can see that it found the TrueSeq adapter, Illuminar's adapter in the read 1. Also, it is detecting adapter for read 2 and also found the TrueSeq adapter, which it is now omitting, which it will trim. And finally, it created the JSON report and the HTML report as well. And it is showing the statistics that it did it in 63 seconds. So it's quite fast actually. And there is also a report like duplication rate, how many bases are corrected for overlap or for adapter. So it's a report on the filtering result. So only 833824, this number of read passed the filter. And reads failed due to low quality, its number is 20. Reads failed due to too many N is 0. And these 166, 156, this number of reads are too short. That's why those were omitted. Okay, so now we can see by writing the command ls what we got actually. Huh? Not this percentage. Did you put all the options? Okay. So maybe there is some alteration in your configuration. 49 seconds. Okay. It depends on the processing. Since we are installing it from which version of Ubuntu is you using? Okay, so maybe you are using 22 and I have the 20. So maybe that can cause a option for a repository. I'm not sure actually. Okay, so have we all got the result for first P analysis? If we write the command ls, we can see the files we got. We have the trimmed first P HTML, the JSON, and the new sequences, the trimmed R1 and R2. If we now want to see you see that I will be using another command now, ZLESS. So these are the filtered sequence now that we have. And for the next portion, the mapping and downstream other analysis, we will use these trimmed files for other analysis, not the main raw files. The output file is this thing, the trimmed. If you remember that we put the small letter O for R1 and the cap letter O for the R2. O stands for output and I stands for input. You can see the HTML file also. Could you see the first P report? It's in the folder, the HTML file. And it says that how many reads were there, then what is the duplication rate, or what was over-represented adapter before and after filtering. So it's a nice way to know your data. And when you know your data well, then you can interpret the analysis, the final product. You can interpret it better. So just go through the reports when you have time and try to understand what it says. And we can anytime discuss that if there is any confusion. So we will stop here now and we'll take a break, and then when we will come back? Oh, okay. 10 minutes. Okay. Okay. In the meantime, you can tell me if there is any question or any confusion. It's the easiest part. The tools we used is not that problematic, and they give a nice result, and also nice result for your eyes, like since it is using the GUI version. Do you want to try this in your HPC account? Do you think you can do it also there in the supercomputer? You can. And if you need to do it remotely, what you can do, and you want to see the HTML files, the GUI format, you can just run the first QC or first P there and download the HTML files to see in your computer, in your browser. I mean, also in the folder, this folder that was created for first QC, you can see that there is a zip folder also. A zip folder was also created, and it contained the images that we were seeing in the HTML file. So if you know the architecture of a web file, you will know that these are the associated images. For example, this is the adapter content, the image, and you can use it for your publication purpose if you need. For example, here is a per sequence quality, per base quality content, etc. Okay. So you can just use the images also, download it and use it for publications. If you need to transfer data from your computer to your supercomputer, do you know how to do that? Upload the file on demand, in the on demand. Okay, that can be an option, but imagine that you do not have the on demand option and you need to use another supercomputer. How will you transfer this? Yeah. Yes, so you need to learn, if you really want to be in the genomics or the bioinformatics section, you need to learn the command line options for data transfer or remote access. So say we want to transfer this folder to my HPC account so that I have this evidence, or I have these files there for further use. Then what I will do, I will use this command called SCP, is a secure copy. So it uses the SSH option also. The secure copy, SCP. Then since I am transferring a folder, which is named first Q, I will write -R. R stands for, I mean, it's saying that it's a directory. Then first Q analysis. So at first I will need to write what am I transferring. So I'm transferring the folder called first Q analysis, and by -R I am saying that it is a folder. Then with a space I will write the address of my destination folder, that where I am transferring this file. So just note that we have some strategy to identify my HPC's location. So for me, this is the username. The username @ I am transferring it to Awen. I don't know if I'm pronouncing it correctly. awens.osc.edu. So this is the your username, then the server name, then you will put a colon, and then the destination of your the address of your destination, which is normally, if I am using home of that computer of my user, then it will be the /user slash, I have the need to have the project name. Okay, you can remember. Okay, just write it down. You have the number in that case. And you can also, I mean, if you need the exact location, you can use the PWD option in your on-demand or other places. So PG is 0 299. Then again, your username. And then slash. I think it's having problem with the internet now. But you can just do it in this way, that username, users, then your PG something, and then slash your username, then again put the slash, then press enter. Hang করে গেছে। Too many things. Any of you could do this? I mean, just users, then PG S0299, then slash your username, then slash, and then press enter and see if it works. Then try it with smaller, dash smaller. So for identifying it's a directory, we can use the smaller option. Okay, we will take a break now and we'll come back again. Okay, 11:20, we can come back.
Did the SCP work for you? Well,
Sorry, Thomas, we could not hear you.
I was just asking whether the SCP worked all right for you getting the files transferred to OSC.
Yeah, I think some of them could do it, right? Yeah, some of them could do it, but my computer actually got hanged. So we will do it after the break again. I hope it won't be the problem. Thank you.